Now let's describe the general structure and function of the nucleus. The nucleus is a cellular organelle. Located in the center of the cell, it contains genetic material, DNA. If you will, it's the cellular headquarters where the DNA contains the blueprint for building body proteins, as we've previously discussed. The nucleus is bound by a double-layered membrane, the nuclear envelope. As you can see in the image here, it's very porous, which allows movement between the fluid inside the nucleus, called nucleoplasm, and the cytoplasm, the fluid that's found within the rest of the cell. That nucleoplasm is a fluid just like the cytoplasm that suspends the contents of the nucleus, the DNA and the nucleolus, shown here. The nucleolus has that name because it means little nucleus. In this area, ribosomes are produced. Ribosomes then have to leave the nucleus to go into the cytoplasm, and that goes back to the reason for the nuclear membrane having pores in it, though there are other reasons as well that we'll look at later in the course. So now we want to explain the relationship between chromosomes, DNA, and genes, because they're all interrelated, very similar to each other, but they do have different definitions. When the cell is not dividing, DNA and protein found within the nucleus form loose, bumpy threads that we call chromatin. That chromatin is spread and scattered throughout the nucleus. However, during cell division, the chromatin collects and condenses to form chromosomes. If we were to zoom in on the nucleus during that condensed time, we would see the chromosomes like this. This karyotype is all of the chromosomes of the human cell laid out beside each other. Zooming in on a chromosome, it looks something like this, where we have the chromosome and in the middle we have the centromere. As we've said earlier, the chromosomes then are made up of DNA. Here's an image of that DNA wrapped around the proteins that ultimately go on to form the chromosome. So the DNA is the nucleic acid that then wraps around proteins and then condenses into this chromosome for cellular division. If we were to zoom in on the DNA, we could see the code that is made up of the cytosine, thymine, guanine, and adenine, which were the bases of the nucleotides that we saw in the last unit. This is the blueprint of how to make proteins, as we've mentioned a number of times. Genes are portions of this DNA then that contain the information to make one single protein. The analogy I usually use is to imagine the chromosome as a recipe book. The recipe book is made up of many pages and many words, but within those pages, each page has a specific recipe that you can ultimately make from the words that are in that recipe. In that case, the recipe is the gene, and then the words are these bases that indicate what to put into the recipe you're making. These genes are inherited traits that are passed on from parents. If we were to look at chromosomes on their own here, we can identify the specific numbers of the chromosomes and the characteristics of them. In humans, we have 23 pairs of chromosomes. Overall, we have 46 chromosomes. And that's because we received 23 chromosomes from mom and 23 chromosomes from dad, such that we ended up with 46 chromosomes. One difference between the 23 chromosomes that we receive from mom and 23 chromosomes we receive from dad is that the 23rd pair are the X and Y chromosomes. A female's 23rd pair are always 2 X chromosomes, whereas a male is 1 X chromosome and 1 Y chromosome. This 23rd pair is what determines the biological sex of the individual. Because the female has 2 X chromosomes, and the male has an X and a Y chromosome, ultimately it is the father that determines the biological sex of the offspring. If the father gives an X chromosome, the mother gives an X chromosome, and you end up with a biological female. If the father gives a Y chromosome, and the mother gives an X chromosome, you end up with a biological male. Let's differentiate between mitosis and meiosis. Mitosis is the process by which cells divide to form 2 identical daughter cells. It's involved in processes like growth. We all start off as one cell formed by mother and father, and then we develop into adults where we're somewhere around 40 trillion cells. How we got there is that we had mitotic division or mitosis of the initial cell over and over and over as we grew. Mitosis also plays a role in repair. So when we have some sort of injury and our body needs to replace the cells that have been damaged, mitosis is the process by which that is done, producing two identical daughter cells for every cell that divides. As I mentioned, after fertilization, all divisions in terms of growth and repair are related to mitosis. But prior to fertilization, how did we produce those sperm, or how did we produce those eggs? The process there is meiosis. Meiosis ends up with two slightly different results in terms of males and females, but each gamete, each sperm, each egg ends up with 23 chromosomes, so that when they come together, they form a new human with 46 chromosomes. Spermatogenesis is the production of sperm. For each pre-sperm cell that a male would have, he produces 4 sperm cells, each with 23 chromosomes. Oogenesis is the production of eggs. For each pre-egg cell that a female has, she produces 1 egg cell and then non-functional polar bodies that are not used in reproduction. As we mentioned earlier then, when the sperm and the egg come together, they produce a new cell with 46 chromosomes, 23 from mom and 23 from dad. Let's go a little bit more in depth in terms of mitosis. Here we have an image of the full cell cycle. There are 2 major phases within mitosis. We'll start with the interphase, shown by the yellow ring at the center of the circle. The interphase is made up of 3 subphases, G1, S, and G2. The second major phase is the M phase, or mitosis phase. It is made up of 4 subphases, prophase, metaphase, anaphase, and telophase. Let's start with the G1 phase. Let's imagine we have a single cell. In that G1 phase, the cell is going to grow and produce more organelles and produce more substances needed for making more DNA. The G1 phase is about the cell getting bigger and preparing then for the S phase. The G of the G1 phase stands for growth. When the cell moves into the S phase is when the DNA is actually copied or synthesized. Once the copy of DNA is produced, the cell will enter into the G2 phase, the second growth phase. This is where the synthesis of substances and organelles needed for mitosis begins. The cell grows and grows and grows until it reaches the M phase, when it has produced all of the substances needed for the mitosis. The mitosis phase, the M phase, has the 4 subphases as I mentioned earlier. Let's take a closer look at those phases. The first phase is prophase. What happens during this phase is that the DNA condenses into the chromosomes that you see here. The nuclear membrane starts to break down and disappear, and the centrioles begin to move to either side of the cell. On top of that, the centrioles begin to produce proteins called spindles that end up reaching out and connecting to the chromosomes once the nuclear membrane has disappeared. We move into metaphase at that point, and then during metaphase, those spindle fibers connect to the chromosomes and align all the chromosomes along the equator of the cell. We then move into anaphase, where the copies of the chromosomes are pulled apart by those spindle fibers and moved towards either side of the cell. You can see that the plasma membrane begins to furrow or indent, and then as we move to the next phase, telophase, it indents more and more until it eventually connects. And in telophase, we have basically the opposite of the prophase. You can see here that the nuclear membrane is starting to reform, the spindle fibers disappear, and even the DNA will start to unwind from the condensed chromosomes that we see in the image here. And then finally, what we end up with are 2 identical daughter cells. Another image for that is we started off with diploid parent cell. The diploid is referring to di, meaning 2, and ploid, meaning chromosomes. So we have 2 copies of all the chromosomes, 23 pairs, or 46 chromosomes, as is shown here. Those cells then undergo division in mitosis to produce 2 identical daughter cells, as we've stated. But in this case, let's imagine that these were primary sex cells, cells that would go on to produce egg and sperm. All of the cells in our body that are not egg and sperm undergo only mitosis. Even producing these primary sex cells, the pre-sperm and pre-egg cells. We have mitosis to do that. But then meiosis is the process of taking those primary sex cells, dividing them into secondary sex cells, whereas you can see here we actually have half the number of chromosomes, 23 chromosomes. That's because in this case, for the sex cells, the chromosomes align along the equator beside each other. So you're pulling one chromosome 1 into the one cell and one chromosome 1 into the other cell, one chromosome 2 and one chromosome 2 into each of the two cells. That happens for all 46 chromosomes. This process is meiosis 1, and then each of those cells undergoes meiosis 2. In meiosis 2, the chromosomes align along the equator, and then the chromosomes are pulled apart, as in mitosis. We end up with haploid gametes, each with 23 chromosomes. Let's reduce the size of this image here. So one other difference between meiosis and mitosis, in addition to the fact that mitosis occurs in all cells but sex cells, meiosis occurs only in sex cells. In meiosis, we end up with cells with 23 chromosomes. In mitosis, they have 46 chromosomes. We also, in meiosis, we can have other possible alignments of the chromosomes, and therefore, if we look at this example here, we end up with sex cells that have different combinations of chromosomes. Here's another alignment prior to meiosis 1 that leads to another combination of chromosomes. This is related to offspring or child production in the sense of, other than in the case of identical twins, no two siblings are identical to each other, because they received a different sperm and a different egg from mom and dad in order to produce them. And here we have the major reason for that variety of genetics, because different sperm and different eggs will end up with different combinations of chromosomes from mom or from dad. And then those different combinations of chromosomes in egg and sperm come together to form the offspring. To relate back to what we saw in terms of mitosis, meiosis 1 also has 4 subparts: prophase 1, metaphase 1, anaphase 1, and telophase 1. You end up forming 2 daughter cells, as we mentioned, each with 23 chromosomes. Those 2 daughter cells then go through prophase 2, metaphase 2, anaphase 2, and telophase 2, and you end up with 4 daughter cells, each with 23 chromosomes. In the case of males, you end up with 4 non-identical sperm. In the case of females, you end up with 4 non-identical cells as well, but it's slightly different. For females, all of the cytoplasm and organelles of 3 of those cells end up going into the other cell, forming the egg. The other 3 cells are actually not the size of these cells here, but are the polar bodies that we mentioned earlier that end up just being sloughed off and killed off, and you form, through this process, just one large egg. Looking at another visual for meiosis and how it leads to offspring biological sex is that if we have a male, the male has a 23rd pair of chromosomes of X and Y. The female has a 23rd pair of chromosomes that are 2 X's. When the male produces sperm, he would have 50% of his sperm having an X chromosome in it, 50% of his sperm having a Y chromosome in it. For the female, all of her eggs would have X chromosomes in them. When those sperm fertilize those eggs, we would end up with offspring that have 2 X's. Those would be biological daughters. And offspring that have an X and a Y chromosome. Those would be biological sons. Finally, we want to discuss the concept of cell differentiation. This is because what we've been discussing here is the sperm and the egg coming together to form one cell. But that cell becomes an adult that is made up of approximately 40 trillion cells. And within those, there's different types of cells in terms of structure and function. So we start off as an unspecialized cell, but we end up with neurons, red blood cells, muscle cells, bone cells, epithelial cells that all look very different from each other and all function very different from each other. How that happens is that unspecialized cell has all the genetics and ability to become any of these cells. But through development, various enzymes are switched on, and then various genes are expressed such that we end up with these specialized cells that have different look and different function from each other. This overall is the concept of cell differentiation, the production of different kinds of cells based on various enzymes being produced and having an effect on the cells in terms of their development.